welcome to yet another revision stream. Because exam season is nearly here. It is just under 4 school weeks until my first exam, which is a terrifying thought, and it means we really need to get pushing now, start focusing on the content which I'm struggling with, start focusing on exam questions, exam response, etc, etc. One of those topics I have put in the top corner here. This is my actual op topic, meiosis. I hate this topic with a burning passion because I've never understood it. Until I had a moment of clarity yesterday, and I might understand it now. I might. I don't know. I might just be kind of making this up, but I feel like I understand it now. So, the way I see meiosis is we have our cell here, our gametes. It then goes under meiosis 1, which is going to lead to 2 cells. Then it goes under meiosis 2, which is going to lead to 4. But that's common sense, okay? That is the basics of meiosis that everyone in their nans knows. What I'm concerned about is the actual explanation here of how it all works. Because yeah, I know that it's 2 nuclear divisions instead of 1. I know it results in 4 non-identical haploid cells. But what does the wording say? Well, I think that the way it works is, to begin, we have 2N, which is a diploid. Then it goes into N, which is a haploid. Then it remains an N haploid. And this is the bit that I think I would get wrong, because I would assume that, let's say the 2N is 48. So normally it has 24 chromosomes, 48 here. That's another thing, actually, is how does that work? Because the way I see it is, when they're replicated, you get things like this. And then when it's a haploid, it's like that. And then when it's... That's the way I see it. So let's expand that a little. Is that not how it works? So, 2N to N to N, you start off with your homologous chromosomes, then your chromosomes, then... I don't know the wording. That's the only bit that I struggle with. So, let us go and have a look and try and understand meiosis so that I can explain it properly and I can understand it properly. Because as far as I'm concerned, that seems about right, but that wouldn't answer many exam questions. Because I still don't know what I'm talking about here. Those are homologous pairs, correct? I think those are called homologous pairs, but I don't know where else you go from there. So, we are going to go on to... There might be a good PowerPoint by my teacher, there might not be. The thing that I'm somewhat confident with is... Oh, and I don't even recall the name of it. Shit, what's the name? There's independent assortment, and then, crossing over. That is where you have 2 homologous chromosomes, they form a bivalent together, they associate to form a bivalent. A chiasmata is formed, which is basically a part of one chromosome breaking off and attaching to the other part. And that creates a new combination of alleles. So, it is saying... Oh, this is year 4 content. What the fuck? I'm not that unadvanced. Um... Oh, so maybe our teacher doesn't have a PowerPoint on Meiosis. Okay. I guess I'm just gonna have to Google this and find out on my own. Oh no, okay. Let's just try and find a nice diagram. Like, preferably a very nice, well-labelled diagram. So yeah, okay, I understand this. So to begin with, before DNA replication, it's this. Okay, let's draw it the way the diagram does, because this diagram seems to be wanting me to draw it a specific way. Let's draw it the way this diagram does. Let's see if it helps. So it's showing line, 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 line. Then the DNA obviously replicates, which means you're going to get 1, 2, 1, 2. That bit I understand. So that's N. No, that would still be a diploid because there's still 4 there. Right? That's still a diploid. Then in meiosis 1, each one is only getting half the number. Then in meiosis 2, they are still getting the exact same number of cells. I should probably I think that is correct. I don't know the wording, because I know there's chromatids compared to chromosomes. I don't know what the difference is. Can we get a little diagram of that, please? Oh, okay, hold up. What we have, okay, this might make sense, is it's saying that the homologous chromosomes are literally just would be like those things. Just 2 pairs. I'm gonna have to Google that to make sure, but then it's also saying that the sister chromatids would be like that. Sister chromatids. What is a homologous chromosome? Homologous pair of chromosomes. Homologous chromosomes are 2 pieces of DNA within the diploid which carry the same gene. Right, I have no fucking idea what that means, to be honest. My brain is a bit mush. Can I get, like, a very simple diagram that, like, a 1-year-old would understand? Okay, I think that's correct then. Yeah, okay. You know what? We're gonna stop there and just assume that we're right, and I'm gonna go into physics math tutor and I'm gonna find some exam questions for this that hopefully mean that I don't get this wrong. because I think that this is it now. This should literally be me understanding it, but let's have a look. Mutation and meiosis. Right, here we go. Please say I haven't answered any of these before. Okay. Complete figure 1. Okay, how are we gonna do this without destroying the diagram? It's showing that the diploid number of... showing how a specific species is produced, the number of chromosomes is shown. So it's showing how in a diploid cell, one has 62, right? Okay, I think I get this. This is just basic math. One has 62, and that would be 2n. One has an unknown value, that would be 2n. That would be n, that would be n. And in total, the species overall has 61 as 2n. So surely, this is just common math, and that's going to be 31, which means that one has to be 30, which means that one has to be 60. Please say that I'm not overthinking this. That is basic, like, year 1 math, right? I love how the mark scheme's not loading. This is, like, incredibly foreboding. Yes, okay, 62 for... Yeah, okay, I got it right. Okay, cool. Cool, that's reassuring. Name the type of mutation that changed the number of chromosomes a mutation in the number of S. Townsendi produced a new species. So S. Townsendi has 61 chromosomes as a diploid. S. anglia has 122. Name a type of mutation that changed the number of chromosomes. It's doubled the number. Is there a specific name? Isn't that non-disjunctive or something? I remember getting this wrong in the actual AS paper because I spent like half an hour thinking of what it was. I was thinking like non-conjunctive, non-disjunctive. What is it? And how does that work? Non-disjunction. Okay, I was close. That is... Explain your answer. The answer it's apparently looking for is... Oh, chromosomes not separated. Okay, cool. That is something very easy, but we can drill into our heads. So it's Is it non-disjunction? Yes, non-disjunction. Non-disjunction is in meiosis, which is easy enough to know. You don't need to do, you shouldn't have to drill that into your head. It should just come. It's where the chromosomes are not separated. So, I might have gotten a mark just for saying non-disjunction. Probably could have gotten a second mark, because it's implied, right? If this species' diploid number is 61, yeah, then if it has 122, then clearly that means something has gone wrong during meiosis, which has doubled the amount. And clearly that would be if this was left. Chromosomes aren't separated, but they go on. Okay, cool. Genetic variation within a species is increased during meiosis by crossing over and the independent segregation of homologous chromosomes. Apart from mutation, explain one other way genetic variation within a species is increased. How many marks? 2. Well, my go-to answer for this question is talking about how fertilization is random, because that would obviously increase genetic variation of offspring. Would that get you 2 marks, Phil? Let's have a look. Yes, random fertilization. And you would have had to specify that it produces new allele combinations. Cool. That was my go-to answer, and it worked. Random fusion of gametes or random fertilization. So, a keyword that I'm already noticing is going to be new allele combination. And that is because fertilization of gametes is random. Cool. So this is 2 bits of knowledge that we already knew, just didn't know fully. So I'm happy we're doing okay. Already done that exam question. Give one reason why trout eggs produced by meiosis are genetically different. That would be, let's just say, crossing over that occurs during meiosis 1 leads to genetically different haploid cells. Oh, you could have literally just said crossing over. Cool. A trout body cell contained 80 chromosomes, so that would be 2n is 80. Farmed female trout are treated so they produce diploid egg cells. Give the number of chromosomes in body cells of offspring produced from treated females and untreated males. So, the males are going to have 80. The females are going to have 160. When they combine together, the females, no, I'm being thick. The females aren't going to have 180. The males are going to get halved. Females aren't going to get halved, so it's 120. See, that's a cool bit of knowledge to keep. If you want to track the number, this is a very useful diagram, actually, the thing I'm drawing here. Look. 2n, n, and overall, your gametes is going to have that many. But I think that's a very useful diagram. So the answer is 120, right? I totally haven't just blown you guys off. 120, yeah. The offspring produced from farm trout are sterile. Explain why. I did just spoil myself the answer, but this should be pretty explanatory. They have too many chromosomes, which means they cannot produce fertile offspring. And the explanation there, yeah, too many chromosomes, or an extra set of chromosomes, so therefore no meiosis can occur. Because they cannot separate equally. Define gene mutation and explain how a gene mutation can have no effect or a positive effect. Okay. Mutation is a change in the base sequence. Change in the base sequence of DNA. This can have no change because DNA is degenerate. So what that means is there are multiple triplets, multiple codons, will code for the same amino acid. Multiple codons will code for the same amino acid. It could have a positive effect if the expressed gene, or if the allele is advantageous. Therefore, I don't know what else I know what it's going to ask for, I just don't know how you would word it. It's saying a change in the base sequence of DNA. So yeah, 1 mark. Genetic code is degenerate, so it will have no change. So you would have also had to specify that this leads to a change in the amino acid produced, or results in a new allele, leading to new allele. So again, linking back to the idea of alleles, genetic code is degenerate. Therefore, okay, this is what we should have specified. It changes the amino acid but has no effect on the tertiary structure. The new allele is recessive and does not influence. Or it's positive because it results in a change in the polypeptide that positively changes. Or may result in increased survival chance. So, I might have probably gotten 3 out of 4 marks there, if not 2 marks, which is disappointing, but that is because we didn't drill in everything we knew there. I rushed that question, actually. If I was in an exam, I would have definitely taken more time to write in detail. The idea that DNA is degenerative, therefore multiple base sequences or multiple codons will code for the same amino acid. That would have got me a mark. Then I would have went on to say, therefore, it would have no change on the polypeptide structure. Also, another fact to consider is, what if the mutation occurs on an intron? That is something I completely forgot could happen. But yeah, introns. So there you go. That's 2 marks for explaining why why a mutation could have no effect. Why it would have a positive effect? Well, that's easy. It leads to a positive change in the polypeptide due to, and it wants you to specifically say, it results in a change in the polypeptide. So you could have easily gotten 4 marks. I could have easily gotten 4 marks there. It has a positive effect because it will lead to a positive change in a polypeptide. That increases survival chances. And obviously what I just said, it would have no change if the mutation occurs on an intron or if DNA is degenerate and therefore the amino acid sequence will not be changed. 4 marks, I would have probably gotten 2, but now I would have gotten all 4. 2 differences between mitosis and meiosis. Okay, difference 1. Mitosis is only 1 nuclear division. Meiosis is 2 nuclear divisions. Mitosis will always produce a diploid. Meiosis will always produce an easier one that could guarantee I don't lose marks. Mitosis will produce genetically identical cells. Meiosis will produce 4 genetically different cells. Easy. Mark scheme better back me up here. 1 division, 2 divisions in mitosis. Yep. The daughter cells are genetically identical. Both are, which is the mistake I was going to make, diploid to diploid haploid to haploid to diploid to haploid in meiosis. So, the daughter cells are genetically identical in mitosis. The daughter cells are genetically different in meiosis. Cool. So 2 marks. Yeah? How many marks was that question? 2. Yeah, cool. There are 7 chromosomes in a spore nucleus. This fungus produces cells at the end of mitosis, called spores. So they are going to be those guys. The number of chromatids present in a spore. And it's saying there's 7 chromosomes. So, wouldn't it just be 14? "28? Oh! No, I'm retarded, sorry. Yes, it gave us the N number. So even if I went back, it wouldn't have doubled. That was the first mistake that I made. The second mistake is on the diagram it showed us 4 spores. So I would have just had to multiply by 4. That is a fucking evil question, by the way. What can you conclude about the movement of chromosomes using all the information? Let's just look at the mark scheme. Independent segregation occurred because they're arranged in different sequences. I would have put that. Crossing over occurred in 3 out of the 10 tubes. I don't know how you would have inferred that. Crossing over is rare and only occurred in 10 tubes. Cool. Okay. Oh, that was asking us to draw a amino acid. Okay, we can still do that. I did just spoil it, but carbon, nitrogen. I like to draw it this way now, actually. I found it easier. Variable R group. Now, this is the difficult bit. I think it's O here, and then an OH there. Please be right. Oh, you could have literally just put COOH. Cool. I drew it more complicated then, apparently. Final question I'm going to do before we head off. Complete figure 2 to show the chromosome content. So, chromosome content after first division. So it's showing chromosome content of a diploid parent cell is 4. So 4 is 2n. So it's showing us this stage here, where you have 1, 2. Well, I'll just draw 1, 2, 3, 4. Then it's asking us to fill out the rest. So after the first division, you're going to have things like 1, 2, 3, 4. 1, 2, 3, 4. Second division, I guess you can just randomly pick, 1, 2, 1, 2, 1, 1, 2, 2. Let's hope that this is right. One long and one short, each made up of 2 chromatids held by centromere. Oh, so I would have had to add a centromere there, just to... Right, well, to be honest, that was a lot. I can't even lie, that was a lot. I hate Meiosis with a burning passion, but I like to think... Hold on, I'm just taking a screenshot. I like to think that did actually make things a bit easier. So, my plan for the rest of the day, guys, is I'm going to go have scran and stuff and chill for a bit. Later, we're going to do history revision, because I have not done a history revision stream properly in a good while. And I want to, because we are actually getting very close to history mocks and history exam as a whole. It's the British paper, the one that we're doing first. So that is my plan. Later on, I'll stream and we'll go over theme 3 for history, which is actually quite a nice one.